Hi, Doc.
Hello, Miss Michelle.
How are you?
We're alive and kicking
We are
today.
alive and kicking. So, I came to the office because I have a couple of questions for you
Hmm.
that I wanted to post out a little bit. So, genetics are a huge part of methylation. We know that.
Well, the understanding of
They
methylation,
on this,
absolutely.
yeah. And I'm seeing all the time on my Facebook page and on different Facebook groups for methylation that people are only really posting one or two genes. Now, a lot of times I'll say, no, there's more than two MTHFR genes, and there's like 22,000 of them.
Hmm.
And I get concerned because people are only looking at like two of the MTHFR genes and they'll throw in a COMT once in a while. So, my question to you is how important is genetics with knowing more than one or
Okay.
two gene SNPs?
All right. Good question.
Thank you.
And it's something that basically people need to really look at and understand. First off, genetics is in its infant stage. It's looking at a lot of different things. There are tons of unknown genes that have not been researched. They know where they fall, they know what their position is, and they know what enzymes they think they pretty much control.
Okay.
But at the same token, they don't have a definitive, this is this disease.
Right.
A lot of what you get from genetics that are out there, and there's different types, a lot of what you get is they're trying to match diseases. You're going to have heart disease because you have this gene or that. And that's not
Yeah.
really that accurate.
Right.
And you're going to have a methylation problem because you have this. That's not really that accurate either. Now, there are different types of genetics. There's what I like to call retail genetics,
Okay.
which is basically we're going to give you a lot of fancy paperwork. We're going to name about 20 or 30 genes, and that's your whole profile, and we're going to do everything from there. That's the Chat GPT version of things. It's very, it's good if you have really no problems. It's okay if you just want to know some basic generalized information, which may or may not be true.
Right.
Now, why do I say that? When you look at the genes themselves, I can't tell you how many times I see people have tons of heart genes, going to have terrible cholesterol, and they got to watch a heart attack risk, and then I'll run a blood work up on them, and the cholesterol's 150. So it's
Okay.
like, yeah, I don't think so. Their LDL is under 100. So why is that? Because genes are never a solo, they're always a chorus.
Okay.
And in being a chorus, you have to look at all of them. The more of the chorus you see, the more understanding of what is actually being played.
Right.
If you look at it from one pinpoint, you really can't. That's like looking up at the night sky and seeing one star and saying that's the universe.
Right.
It's just not. Then you get to higher level genetics, and there's a lot of companies that are doing that too. The more in depth you get, the better it is. Started off back with Ancestry
Yeah, I remember
and
that.
all of those. They were basically running a lot of the gene SNPs through that, and they kept changing from the different chips that they would use, which changes the information you get. Now, depending on which ones you're running and how much you want to spend, is how much you get. I particularly like what I'll call a mid-range one. The company Genome and All makes an incredibly good genetic profile, as opposed to the ancestries, as opposed to some of the genome, which we call it doctors' datas and things like that. The ones that are running it, Genome and All is one of the better companies. They give you a massive number of pieces. Now, is it for everyone? No. It's a higher level piece that's going to show you more information and more genes.
Right.
Is it great for practitioners? Absolutely. For retail people, if you've got somebody who wants to see your genetics, or you're going to want to find a practitioner, Genome and All is better to go with because it gives much, much, much more information.
Like how? Like what is different between like what information do they provide?
Well, they provide more genes and more correlations. They also provide a tremendously high amount of biochemical pathways, which matches with blood workups, amino acid scans, and things of that nature. So when you're looking at a genome at all, you're looking at a lot of different parameters, whereas a lot of the other ones will turn around, and I'm not going to mention them all because they'll really get angry believe me. They're doing more things like this particular amino acid does this, or this one does that. And they're discounting things depending on what research is out there. What you have to remember about genetics, and this is the most important thing for people to realize, when you look at a gene and you see a nice fancy thing of what it does next to it, that doesn't mean that's what it actually does. That means what's been researched. Some guy in a lab, some lady in a lab sat down and said, we want to do this, and we want to see what this gene does, and they do that specific testing for that specific piece. It's a one-on-one system. They want to know one bit of information about it. Mm-hmm. It doesn't mean that's all that the gene does. Mm-hmm. It just means that's that one bit that they investigated, they looked at, and they've proved it, had it peer-reviewed, and it's published.
Okay.
So that's mostly what you're seeing a lot of times on these genes. Mm-hmm. But that doesn't mean that that's a broad spectrum view of that gene. That means that that's one specific set of researchers who have looked at that gene and say, this is what we found it does, and we can prove it. And that's great. You need guys like that, and you need gals like that to keep doing that till the cows come home, till we know everything that's going on there.
Right.
But it's not the full answer. When you see a lot of genetics will tell you you have an MAO problem. Well, is that MAOA? Is that MAOB? Okay, is it upregulated? Is it downregulated? They don't really tell you a lot of it. They're just saying MAO problem. Okay, mm-hmm. That's not accurate enough. How many MAO problems do you have? 1 out of thousands? Or do you have 100? If you got 100, that means you're definitely going to have a bigger problem than if you have 1. Mm-hmm. So you have to start looking at the genetics. The more information you have, the better it is. If you're somebody who is just looking for what are the best mid-range genetic ones and doesn't want to spend $1 million, but wants to have a really good, gene set for a practitioner or a doctor or someone who is going to read these things for you, mm-hmm. Genominol is the best one to go with.
So you were saying before that and I'm staring at 2 different genetics.
You can show my screen. There's
no names on it.
Yeah, there's nobody there.
So
this is a Genominol screen. This is what used to be called the Ancestry Prometheus screen. Okay. Mm-hmm. Now, they're both set at this particular moment on MTR. If you look at the Prometheus screen, you see that the Prometheus screen here has all these MTRs,
right,
and they don't tell you anything.
Okay.
They're all blanks.
And for you as a biochemist, you need to know.
Well, then you come over here, you see there's 3 MTRs that they're talking about, and you notice that their variants are there. Mm-hmm. Well, the difference is that on this one, they're not these numbers because the chips are different. Mm-hmm. When you do genetics, you're looking, and the chips that you're looking at provide you either with all of these numbers or all of those numbers. If you're not looking at a broad spectrum of ones that are biologically active, you're going to get a lot of blanks. Mm. You're going to get a lot of unknowns. Now, that's not to say that Genominol doesn't have tons of unknowns too. They're going on in research that's published and put out there, mm-hmm. and how everybody is working with that. So this makes it different. As a practitioner, looking at these, you get a better understanding with Genominol, because I've found that they deal with neurotransmitter production as well as breakdowns. They give you a larger understanding of the physiology of a person. Combining a Genominol with a blood test as opposed to, let's say, an Ancestry with a blood test is very different. Mm-hmm. You get a lot more information about what's happening real time with the Genominol than you would with an Ancestry.
But you could still do work with Ancestry.
You can work with every genetics.
Right.
It's the level and depth of work you're looking to do.
Right.
If you're just looking to basically find out if, you've got an APOE4W, then mm-hmm. yeah, you can do that, but that's not really going to tell you whether you're going to have Alzheimer's. It just tells you that you have a higher risk of Alzheimer's. Right. Now, if you look at it and you've got 10 APOEs, and you've got high cholesterol in your bloodstream, and you've got all these other sections going on at the same time, now you've got a picture that, I got to start doing something about that. Mm-hmm. But if you're going to do it from a generalized basis of just looking at 1 or 2 genes, that's not enough.
Now I have a question, because I always have questions.
Always.
So how how safe is it for people who are asking people,
I already know where you're going with this.
Okay.
We talked about this one
because
before.
it scares me.
People who get an idea of one gene because they ran an ancestry.
People, lay people.
Or they ran a doctor's data or one of those type of genetics. Okay, if you're a lay person and you look at it and you say, oh, I've got this gene, that doesn't really mean anything.
Okay.
It's not something you can treat yourself off
Right.
of.
That's what I.
Having an MTHFR, if you have 2 of them out of the 22,000, okay, you really need to know what is around that MTHFR.
Right.
If you don't look what's around that MTHFR, you're not going to get an understanding of whether that MTHFR means something or doesn't. I've run genetics on literally hundreds and hundreds of people at this point, up into the near 1,000. And when you look at all of that, what you find out is that genes are very you've got to really analyze them and know what you're looking at to come up with any kind of answer. When you look at an MTHFR gene, I've got tons of people who have an MTHFR gene. They eat fish, they eat rice, they don't eat fish, they don't eat rice, and they're fine. They don't have a problem. So what does that mean? Does it mean that the MTHFR is lessened in function with them? No. It means basically that the downwind effects of the MTHFR are not in play, and therefore doesn't bother them at all. They don't have any functional problems with any of those things. Those people are real lucky. There are a lot of genetic variants out there that people have, okay? I just did one the other day. There's a disease called alpha-trypsin, okay? And what it is an enzyme, and it controls a specific type of function in your liver and lungs. Not to get into the insanity of it. There are 3 major genes that people see for alpha-trypsin disorder. One of them called 1303, all right? If you have that gene, you may have an alpha-trypsin disorder, or you may not. But it's there, it says, oh, you've got alpha-trypsin. If you go and get the enzyme tested, a lot of people don't have it.
Right.
Okay, because there's other compensation factors. So if you have major problems and you want to use your genetics to get at that, then you got to do something higher level like a genominal. If you're just looking to see whether or not you can eat kumquats, on a regular basis, then yeah, you can get away with the lower level retail genetics. If somebody's selling you a bunch of genetics and they've got 20 of them on there, okay, and they say this is your nutritional personalized profile from 20 genes, no, that's basically very lightweight, and so long as you don't have any problems whatsoever, you're okay. If you've got problems, those kind of systems are not going to work.
All right, so my last question. Thank you for talking to me today, by the way. I know you're very busy. So my last question is, I'm sitting here. I've seen so many genetics over the years that, I've been with you and then writing the book and all that stuff. And I see the amount of writing that's next to the genes. How have you been able to customize your?
Well, I don't customize.
Okay.
What I specifically do.
It's customized to me. It's individual programs.
This is what people hate. Well, you can't do it any other way.
Yeah, well, that's what I'm saying.
What I do specifically is I go page by page, which is why it takes me excruciatingly long to do these. Right. I go page by
That's why
page.
I haven't seen you since August.
Yeah, pretty much. The genominal, for instance, there's about 1,200 pages to 1,300 pages of genes you go through. And why do I do that? Because I approach it from a biochemical standpoint, and when you're approaching it from a biochemical standpoint, I'm not looking for the magic gene that's going to give you a heart attack. I'm looking at the 50 or 60 other genes that create plaque in your system that can lead to a heart attack. So you have to view it from a different standpoint. I'm viewing it more from the specifics of this is what it is. This is why it takes so long to read.
And it makes the programs that you do more concise.
Well, it makes them more accurate to the individual because I'm also combining that with blood work.
Right.
Okay. And there's different types of blood workup depending on what the system problem is. For instance, you can have a standardized regular blood workup, okay, your comprehensive metabolic, your thyroid, and all of these. That matches up well with the genes, and it tells you a lot of stuff, okay? Then you get into like genomic, and you've got stuff that's really very esoteric in there. Okay, you can look at your neurotransmitters. You can look at neurotransmitter breakdowns. You can look at amino acids.
Oh,
Now you
all.
can see.
Yeah.
Now you see all of the different pieces that the amino acids are doing. You see what the enzymes are doing, and you get an idea of how they mesh. That really tells you about that individual. Okay, which is the only way if you're going to do nutritional work on somebody who has any kind of specific disease states, or we'll call them metabolic malfunctions. People have to remember the genes are not going to tell you only about your diseases. That's a medical interpretation of genetics. Right. Medical interpretations are based on you have these 10 genes, okay, and you have these symptoms, and that matches this blood part of the blood workup, and therefore you have this disease. Okay? Those are people who have a disease and they're trying to figure out and verify that that's that so that they can treat it from those standpoints. Right. When you're dealing with it from a biochemical standpoint, what you're doing is looking at a person's system like a big roadway, okay? Where are the traffic jams?
I got a lot of jams.
Exactly. Where are the traffic jams? Where are the things that are combining down to one lane? Where is it a 6-lane highway? How do you route around those jams and those one lanes and get to a better metabolism so that the person has a better quality of life and functionality? That's really what we're doing when you're dealing with that.
Just out of curiosity, can you name all my jam-ups?
No.
Come on.
No,
try.
I can't. There's too many in there. You've got pages. Remember, when I did your genes, you came up with something like 30 pages that I cut and pasted out of there. Okay, but that's also your genetic history from your family.
I'm special.
And well, you've got the genetic history from the family also. That's really got some things that are passed along. Now, just the simple fact, and I'm going to say it.
Say it.
The ovarian failure.
Yep.
Okay, it's in your family. It's in the female side of the family.
Yep.
Okay, all of that has a number of different gene malfunctions to it. It's not just one gene.
Yeah.
There's not one gene that says, oh, by the way, ovarian failure. There's dozens of them. There's a CYP17A1, there's a CYP19A1.
There you go.
There's all of these different pieces that go in there, okay, that affect how your hormones play out, how your healing and repair plays out. That's not to mention that the methylation also affects that by over-stimulating immune system activity. Okay, so there's a lot of different things that go into this. People who are trying to diagnose and fix themselves on one so gene basis they're going to hurt themselves in ways they don't even know. And do I mean it's going to kill them? No, but it's going to cause them more problems and make them worse in a lot of ways.
Yeah.
How many people come into my office and say, "Oh, I put in SAMe, and I couldn't function for days." Yes. Because you don't have the right genetics for that. Your downwind functions from your methylation is a problem. Doctors that are just tossing in methylation. I have a baby, okay, who basically was born and has an MTHFR. She had hydrocephalus when she was born, which is water on the brain, which they surgically did an amazing job and basically cut a little hole into the brain tissue and allowed it to drain. And so far she's doing well with that. Okay, but she had an MTHFR, and the doctors decided that the MTHFR, well, I think it was a 974, one of those, someone that nobody's really going to hear of. What they did is they punched the MTHFR into the genome database they had, and it popped this one out as part of her issue. So they started giving her massive amounts of methylfolate. Now, does that mean the methylfolate is the answer there? No. The methylfolate, this child was basically had failure to thrive, failure to function, couldn't sit up, had no motor functions whatsoever, couldn't roll over, was failing in every aspect of it. So what we looked at, and we found that the baby had CBS gene and CTH.
Did they run a genome at all, or they just ran?
I ran a genome.
Oh, you ran, okay.
Okay, I was called in because the parents were like freaking out, and basically we looked at it. and I looked at the CBS to see, so we started to do different things. Now the baby is sitting up. The baby has spoken her first words. She said, ma, and she said, da. Okay.
For Doc? No, I'm just
For
kidding.
dad. She basically is able to sit up on her own now.
That's amazing.
Can crawl and grab things. MTHFR is a gene. Yes. It has a lot of important functions. Yes. Is it methylation chemistry? No. It is basically folate chemistry, which aids in methylation chemistry. It is not methylation chemistry. The actual methylation chemistry that functions most of the time downwind is SAMe. Now, to get to SAMe, MTHFR helps in the recycle from homocysteine. Yes, it does. But dietarily, the more methionine-based foods you eat, the more methionine you're going to have to make SAMe. That doesn't require the folate portion. What does that require? A gene called MAT1A. The MAT1A gene needs ATP to be active, and when it's active, it then turns methionine into SAMe. That SAMe is then your main methyl donor all over the place for a lot of the functional pieces of your system. So if you're not looking at the diet, how much methionine is coming in from the food, okay? If you're not looking at the MAT1A gene, the MTHFR doesn't really tell you anything. It just gives you an idea of how you're cycling your B12 into methyl B12 so that your homocysteine turns back into methionine.
Right.
That's really all you're doing. It doesn't tell you about the CBS gene. It doesn't tell you the CTH gene. It doesn't tell you whether it's going down to see and make glutathione correctly. Okay? And to give you the idea of the complexity of just that, the CBS CTH genes chain homocysteine into cystathionine and then eventually into cysteine. Cysteine then combines with glutamine to form glutathione.
Right.
And then the glutathione forms its own circle of reduced and non-reduced forms. Okay? But if you don't have any glutamine, even if you fix the CBS gene, you're not going to make glutathione, and that means your immune system is going to have major problems, anti-tumor, antiviral, all of those. So you really have to look at the whole system.
And you really need like a very
That's
thorough.
why when you look at the genome at all, okay, the genome at all will show you, okay, yes, here's a GAD1, here's a GLS, here is a whatchamacallit, ABAT. It'll tell you all of the glutamine pathways. Once you get an idea of that, and then you match that to a blood test, you know whether the person has enough glutamine or whether they need glutamine.
Right.
Now everybody, glutamine is an excitatory neurotransmitter. Yes, it's also 40 million other things. So you can't look at it.
The genomadal genetic, that'll show you the 40 million other things. So
It shows you, well, not 40 million,
I'm
but
saying,
most
so
of
to
it.
speak.
Yeah, it shows you large blocks of the metabolism because it focuses also on metabolics, which is one of the great things about genomadal. It tells you the metabolics. It tells you what pathways are having issues. Now, nobody really knows where specifically that individual gene works with that enzyme in this particular body.
Right.
Nobody's really sure of that yet. They're still working on that. But they haven't quite gotten that down to a science yet. They're doing a lot of different things, and there's a lot of hope for genetics and everything else coming about.
Well, I remember when I first started with you when I was 24.
Right.
You didn't even
They weren't
you didn't even run genetics. There was
Well, they
nothing.
didn't have genetics. It wasn't there at that time.
Yeah.
We were mostly focused on amino acid scans, neurotransmitter scans, metabolic pathways. That was the main way that I started functioning. That takes me back to the early days of the Orthomolecular Academy of Medicine and Dr. Koonin, Abramonoff, and Linus Pauling, that whole crew. Koonin, I actually had the pleasure of meeting in person. And we sat down and he was giving a lecture and I attended. And afterwards we had a nice conversation. We spoke for about an hour and a half, standing there just back and forth about genes, going over serine and what serine does and doesn't do.
Yeah, I love serine.
And well, serine has a lot of stuff, but see, you see that in genome and all.
Right.
There's a set of genes that controls serine's function, and it shows you that. Okay, and that's everybody focuses on the serine glycine shifting for basically the difference between SAMe and S-adenosyl homocysteine, and everybody looks at that. But serine is also one of the major ones that remove homocysteine down the pathway into cystathionine. So there's a lot of different uses depending on what cofactors you're putting with it. Okay. When you're looking at genominol, it gives you those cofactors. For instance, the serine mechanism we're talking about needs B6 in the form of B6PO4. There is a gene called NBPF3, which controls how much B6 you hold on to in your system and how much washes out. Now, if you give tons of serine and you got an NBPF3 gene and you don't give a lot of B6, you're not going anywhere. See, this is where the complexity of this becomes much, much more than an average person's going to get at a chat GPT or looking at a few diagrams from a textbook.
Right.
The diagrams from the textbook are great. They are the pathways you need to know, and you need to understand those pathways. But when you get to the clinical level, there are no two people on the planet that use the same biochemical pathways in the same way. So they're all unique.
So would people get like an ancestry and a genominol depending on who their doctor is? I mean, I know I get the genominol because it's Olivia and I need a genominol.
Yeah, no, there's a lot of different systems out there. Okay, there's 10X, there's tons of systems that are out there doing different things. As a consumer, as a retail person, or someone who's sick, you have to decide what the level of illness and everything you have is. Okay,
and that,
if it's very generalized and you're a sports person and you just want to know how much creatinine your body can take before your kidneys give out, okay, then you want to do something that's going to be geared towards more athletics.
Right.
And those are those gene sets. Okay, every company has a specific set of genes that they focus on. Okay, some of them are more medically oriented. Genominol is biochemically and medically oriented, which is what makes them a much better system. Now, it's also, and I'm not going to lie, I'm going to say it out loud, their pricing is a lot better than most out there to get the level of information you're getting.
that's good.
Okay, they're a lot cheaper than a lot of the other ones. You see them at 700, 800, 1,000. You don't keep your own genes. They use it for their research and everything. Genominol isn't like that.
So that's another thing. Where, everyone's fearful of where the information goes. So in genetic testing, when they send it, when they send it to you, do you know, is it lost somewhere?
Well, for me, when I look at genes, I usually am looking at the end result of things. I don't specifically run genes.
No, I know that.
I tell people to do that.
I know. But where
Two, the companies that you deal with, it's there on their website what they do with your genes.
Okay, right.
Okay, you have to really know.
I haven't had my genes done in a long time.
Ancestry puts them into a database, and they use it for location and finding friends and everything else. So your genes are in their database, okay? So same thing with 23andMe and those style of companies. They're retail genes. They're more for looking at your family histories and what genetics you have there. That's helpful in some ways, but as the big BRCA gene scare came about and everybody started, cutting off their breasts and things because they had the BRCA genes, it became more evident that that's not the way you use genetics. There are hundreds and hundreds of BRCA1 and BRCA2, and maybe a handful of them are definitive cancer genes.
So the company holds your genetics, so they're not
Some company holds your genetics. Some of them lock your genetics down. There was a new company I was looking at that just came out. They're about $1,500 for a scan. Okay, you don't get to look at your genes. You only get to look at pieces that they'll release to you. Okay, they lock your genes down. They put them into a vault and they lock them down, and that's it.
And what about genominol? They lock it down?
I don't know if genominol. I'm pretty sure they don't, though. Okay, things like ancestry and stuff, they do. You have to contact the individual company. What I like about genominol is that they're straightforward, direct company. They do your genetics, they give it to you. If you want their analysis, they'll provide you with an analysis, which is not a bad way to go. I mean, it'll tell you lots of things, but it's not going to be an in-depth analysis that a practitioner would use.
Right.
And what I do is I go through all of these pages and basically look for things that are relevant to that person, their blood workup, their amino acid scans, their neurotransmitters all together.
Okay.
That's the only way you get a real accurate thing of what's going on there or not going on.
Okay.
Okay? So Genome and All, as far as I'm concerned, is one of the better companies I've seen in a long time as far as wealth of information, useful information. I mean, there's a lot of things in your genes that are not useful. And I'm not going to say that Genome and All doesn't have that.
Right.
There's a lot of medical conditions and things that are in there. As far as a practitioner goes, if you really want to know if they have this specific gene set for this type of, genetic malfunction, yeah, it's great for that too. But they're also great for the amino acid work, for knowing which amino acid pathways are damaged and jammed. That, for anybody who's doing orthomolecular or medical or biochemical molecular treatments, tells you right off what pathways you're going to have problems with. That helps with dietary controls.
Yeah,
A lot
totally.
of people look at diet and they look at it from the standpoint of fats, proteins, carbs. That's retail, okay? And there's a point for that. Then you got to step up, and you've got your athletes who are looking at specifically foods that build body, do this, do that. That's another sin. Okay, there's a lot of different ways to look at it. And then you look at things from the molecular standpoint. By adjusting foods, you're adjusting the amino acid balance coming into the person, which automatically adjusts their metabolism. Why? If you have a gene that is blocking this specific amino acid set from being totally utilized, and you lower that down in the diet, that gene is going to be less likely to express and give you a problem.
Mm-hmm.
If you push it up in the diet and you overload it, that gene is probably going to malfunction, and you're going to have a variant activity blown out in your system across the board. So when you look at diet, you have to get into more than just, is this healthy or not healthy?
Right.
The term healthy or unhealthy, clean, these are great catchwords to sell things. They're made up by marketers. They have nothing to do with reality as far as the chemistry of your body goes. Okay? There are people who have such good genes that they can eat McDonald's every day of the week and it doesn't bother them at all. And then you got people who walk through the the restaurant to go to the bathroom and they're practically passing out.
Right.
Okay, so you can't look at the food from the standpoint of healthy or unhealthy. Yes, is it better to be, quote unquote, use the term clean, have less pesticides, dyes, and things of that nature? Absolutely. That's a wonderful way to go. But that's not everybody's definition of clean. And that's not everybody's understanding of the word clean.
Yeah.
So you have to look at all of those things.
Okay.
When you're getting into genetics and you're getting into actually looking at different aspects of your own physiology, it's how deep you want to go. If you want something that's mid-range, that's not super expensive, I like Genome and All for that. If you want something that's very generalized, that you can pop in a few things and say, ooh, I have this, I have that, okay, then the 23s and the Ancestries and those are all really wonderful for that. There are a lot of higher level companies that the doctors are using and that are medically based that are very expensive for average people and probably out of their range medically.
Right.
Okay, so those are the kind of things you don't want to get into unless you know that your family has a specific gene set. I mean, if everybody in the family has a specific condition, then yeah, you want to go medical, you want to know.
Right.
Okay, but if everybody doesn't, then you're suddenly coming up with symptoms that nobody in the family has or that are in the family, but they don't seem to cause illnesses and they've been given clean bill of health, then a mid-range genetics is where you want to go, and the Genome and All is the way to go for that.
Right. Yeah, because I just kept seeing all the different types of genetics on Facebook and what people are talking about, and I wanted to ask you like what your thoughts were.
And it's a good thought because people are going to get lost in the sauce. Unfortunately, nutrition and things out there are controlled more by who's selling what
Right.
than they are about what they're actually doing. It's difficult to do medical one-on-one research with a nutrient since nutrients do dozens to quadruples to thousands of things in the human system. It's hard to say, oh, we're pinning it down to this mechanism at this one time, and that's the rate limiting step for that specific. It's very hard to do that with nutrients, which is why nutritional chemistry is very difficult, and at the same token, is probably one of the more important things. In medicine, you're not looking for that. You're looking for these numbers mean you have this disease, which means I give you that treatment. And we need that. Okay, I mean, you get bubonic plague, you want to really know that you got bubonic plague, you want to know that this antibiotic is going to work for you. Okay, but if you don't have
Unless
that,
you're me. No.
well, no, you want to know that too. You want to know which antibiotic's not going to work, which one will kill you
We
beforehand.
don't want to go there anymore.
Exactly. See, you've got to know that.
Yeah.
The cytochrome P450s give a very good idea of what chemicals and things you can ingest. They're part of the phase 1 detox, but that doesn't mean phase 1 detox is all cytochrome P4. That's not the way it goes. And there's no guarantee that you're not going to need the NADH to cycle the cytochrome P450s and reconstitute them, because you've got a slow cycling system, but you've got a normal production. So there's a lot of things that go into this. And the more down you get into the weeds, the more intensely complex this becomes. For people who are trying to do their own genes and their own genetics and everything else, I'd say if you don't have any problems and you want to do that, have at it. Enjoy yourself. Use your chat GPT. Go through the genes and come up with all kinds of info. Remember that the information you're getting is on a research from a single researcher who's done a paper. If there are multiple answers in the box, then yes, there are multiple people doing different things. But you'll notice anytime you do genetics and you look at a big panel, like a genome of all, you'll find on one page it says, don't eat broccoli. You'll die if you eat broccoli. 3 pages later it says, you must have broccoli. If you don't have broccoli, you're going to die. Okay, so how do you look at that as a non-practitioner, somebody who doesn't understand? You're going to go out of your mind. You're going to say, I should avoid broccoli, I shouldn't avoid broccoli, I don't know. But the answer comes down to is, which gene is really expressing and which secondary genes are expressing that make the broccoli either detrimental or not. Broccoli happens to be high in sulfur. So if you've got a CBS gene, okay, then broccoli is not a friend. Will it kill you? No, but it will probably make you pretty sick. Okay?
If I eat a lot of it.
Yeah.
Yeah, I don't eat a lot of broccoli. I don't eat a lot of cauliflower
because you've gotten to the point where your genes and your genetics are down to a baseline level, and you can get away with little insults here and there like you did when you were a kid.
Yeah.
But you would sit down and eat broccoli, fish, have 2 glasses of wine, my guess you'd probably be in the hospital.
I'd be clawing at your office.
Yeah, so
All right.
it comes down to things of that nature. But if I had to guess, if I had to say anything, for anybody who's looking to get deeper into their genetics, looking at a bigger and more intense amount.
Yeah.
And
okay, well that's cool.
Genome at all would be a better way to go than just a simple ancestry or 23.
I guess it's like, different doctors,
Doctors all have...
No, what I'm saying is like and forget doctors, universities, like this university is the community university, this is the higher college, and then you've got the Harvards.
The doctorates.
The doctorates, yeah.
Yeah, see,
right.
Genetics for each doctor is also what they're looking for, okay? There's no school where you go and you get a genetics degree to understand how to apply that clinically.
Right.
That's not happening yet. It's getting there, but it's not happening yet. You've got a lot of geneticists who are looking at different issues in there that are getting to that point, but they're focusing on one segment here or one segment there. Okay. You've got doctors who, neurochemists and psychologists who will concentrate more on neurotransmitter genetics. And then you've got doctors who are dealing with detoxification, and they'll deal with more detoxification and methylation genetics. So everybody's got their little thing. The bottom line you have to remember is there's no separate system. It's all in the same thing. It's one body. So all the systems are at play. You can't focus only on one and think that you're going to have your answer.
Yes.
You have to focus on all of them. Yes. Otherwise, you're going to end up with problems.
Biochemically, too.
Well, biochemically, remember, why I like genetics. Where I started, my undergrad was in chemistry. So where I started was chemistry. Okay? What I was looking at is there was an interesting PhD in Stony Brook University Hospital, and he wrote a treatise, and it was about that thick.
I'm sure you read the whole thing.
Okay, oh, I love that. And what he did, his thing was looking at blood workups, because it was a teaching medical hospital. So he was looking at blood workups, and he was looking at the biochemistry of what makes a blood workup specific value change. that was where I started. I looked at that and it was like, oh, this is different. Then I got involved with the Orthomolecular Academy of Medicine through a couple of different sources, Huxley Institute here in the States. Okay, Orthomolecular Academy of Medicine was mostly Canada. But I got involved in looking at that. And at the time, Koonin, Abramonoff, and Pauling, they were all looking at methylation as being the causality factor for schizophrenia. So that's where a lot of this early work comes out of, is that they were trying to say, yeah, fault in the methylation sequence causes schizophrenia. Okay? As they went along, they realized, no, it's a little more complex. And they kept looking at the complexity getting worse and worse and worse until they realized there was no one causality.
Right.
I see now a psychologist, a neuropsychologist who does genetics, and I'm not going to say his name, but he came out with the statement. He said, maybe we shouldn't ask our patients if they're schizophrenic. We should ask, how schizophrenic are you? Because everybody's got schizophrenia genes. Everybody's got all of those.
Oh, that's, yeah.
So-
That's really, yeah.
So when you look at all of these different pieces, the key to remembering it, and this goes for everybody out there who's looking at their genes and everything else, it takes more than one thing. It's not one gene. And just because you have one gene does not mean that that's expressing, does not mean that that's causing you symptoms, and does not mean that that's going to cause symptoms in the future. What it means is you have one variant. Look at all the other variants and analyze all of that, and then you'll know whether that variant has importance or not.
Well, thank you, Dr. Dulin. That was a fabulous ending.
Well, I just figured it was the way to go.
Awesome. Thanks again, Doc.
A pleasure.
Bye, everybody.
All right. Bye-bye.